going to do today is extract DNA from strawberries, okay? It's a pretty easy procedure, works pretty well. But what's going to happen is you're going to bring in pairs of 2, and on the back table you'll meet me, and one of you will get a beaker like this off the table, take it to your lab station, find a funnel, and you'll also take a piece of cheesecloth. You can see mine stained a little red because I used it this morning. The other person will come up there, so I'll only have 11 people up there, and I'll give you a bag with a couple of strawberries in it, or a great big strawberry, and then I'm going to put some buffer solution in this bag. Then you will close the bag, and you will crush the strawberries in the buffer solution. Carefully. You don't want to go smashing on this bag and break it and have the stuff go all over. So you don't want the bag to break. So in the buffer solution, we're going to crush the cells of the strawberry. Why are we doing that? What's going to happen? The buffer solution, by the way, is a soap. It's a soap, okay, basically. And it has some salt in it, too. What will the soap do you think? And what are you doing when you're doing this? Where's the DNA in the cell?
In the nucleus.
In the nucleus. How are we going to get it out of the cell? Yeah, we're going to break the cell walls. And then, what's inside the cell wall? Huh? Well, what? Remember, right? The cell membrane. So what is the soap going to help you do? What's the soap? What is the cell membrane made of? Remember, a phospholipid? It's a double layer, like a sandwich, a phospholipid. And if you use soap, say you get your hands greasy at home, what do you use to get the grease off?
Soap.
Soap. So the soap's going to help you break the cell membrane and get the DNA out of the cell. Okay? Then after you do that for about 5 minutes, you'll pour that liquid through the cheesecloth to strain out the pulp, and the DNA and the rest of the fluid will go into the beaker. Then, when that's done, you let me know and I will bring some alcohol to you. It'll be isopropyl alcohol, and it's cold, and you'll pour that into the beaker slowly and watch for a while, just a few minutes, and it will rise to the top and form a layer, and you will be able to see the 2 layers. Once that happens, you will see in the alcohol layer that the DNA is forming fibers, and you'll be able to see it. Then you'll be able to take a cotton swab or Q-tip and reach down in the beaker, just into the alcohol layer, and slowly swirl it around, and it will pick up the DNA. You'll be able to pick it up and look at it. So it's pretty simple. All you have to do is carefully read and follow the instructions on the lab handout, and then you can work on answering the questions. When you're done with the lab, you have your strand of DNA to tape together, and I think a reading guide to work on too. Okay.
They actually don't smell weird.
It smells good.
Smell like strawberries, right?
Yep.
It's just a little soap. All it is dishwashers, not dishwasher.
Not yet.
We didn't put that in yet.
Dish soap that. When you
Were
put
we
it in
supposed
there,
to put it in right away?
no, you don't have to. You
I
can
was
do it
gonna
either way.
say.
I think we got it.
You
Okay,
can do it
Hunter,
either
I'm
way.
ready.
Move.
Okay.
Not
Well,
yet.
now I'd put your soap in there.
We need another
Open
like
it.
minute or 2.
Yeah.
I was smart enough to do
I
that.
don't want to
Is
do
it
it.
leaking?
Smells good.
Yeah, maybe just leaked out the seal. So how's it working? It looks like we got layers there, doesn't it?
Yeah.
Cool.
Hey!
How's the crushing going?
Good. Good.
done.
Ready to dump it in there.
Should we spread this like right across, or should we roll it up in about?
Well, just get it in there a little bit. You just don't want to pour it in all at once, because if you do,
Oh, then, oh yeah.
Look, stinks.
there
Ready?
you go.
I want to drink it.
Yeah, it strains pretty fast.
Yeah, it does.
And then just throw the pulp away and rinse off the cheesecloth. We'll save it for the next group.
She's happy, but she doesn't want her to like find out it's a joke.
How's it going? Crushing it up, huh?
It's taking forever.
The longer you do it, the better it'll get.
Zoom up on your face.
I'll get it for you. So we're gonna get some more alcohol.
Wait, I want to be in it.
Okay, you're in it. There's your DNA.
Smile.
Smile. Now what are you gonna do with it? You
Oh, I thought he was taking a
you gonna
picture.
touch it?
No, that's freaking nasty.
No, you can touch it. It won't hurt you. After all, if you eat strawberries, you're eating that, right?
I'm not eating strawberries ever again.
Well, if you eat an apple, you're eating it. If you eat meat, you're eating it. Everything you eat
What do
has
we do with
DNA in it. You can throw it away if you want, or you can keep it and take it home and show and tell.
Put it in here.
I can't get it out.
Keep it in here.
Oh, okay.
Swish it around a little bit. Now you can pick it up. Whoops.
Wow, what a queer.
There you go. DNA.
Smile, Lindsay.
How did you like DNA?
You're a crabby little girl.
Oh, this is a really good apple.
I like doing it.
So Lindsay likes her DNA.
She wants to eat
I like my DNA
You
too.
like your DNA too. Yeah.
This is so good.
Isn't it interesting that that little gob of snot
It
is
is
what
so
tells you what to do?
Snot, that's sick.
Probably. I gotta watch this DNA construction here, this DNA model.
Me and Muggy are DNA.
So we're putting together the sugar, phosphate.
Wait, I thought it was sugar and proteins.
It's a phosphate, PO4 group.
They told me it was
It's a
protein.
ribose sugar.
Alright. Now,
And what are these things called?
what?
The guanine and the adenine,
And this
the
calcium.
C and the G and the A and the T.
You know, you already know.
Yeah, you know.
You know.
What, do they hook together like this or something?
You know.
Oh, yeah.
Yeah, that's a good hook together way.
Is that
Very good. They fit. They're cut to fit. Sort of like
So just
making
boom
jeans.
like that?
Yeah.
Alright.
And then you got to put them across here. Now, do you remember what they connect to? I think it's the
A connects to T, G connects to
right. And then what do those connect to on the phosphate and sugar backbone?
What?
The sugar, right? They connect to the ribose sugar, right? Why do they call it deoxyribose? You know, it's because it's deoxy.
double helix or something.
It's because it's deoxy. Let's see how this molecule is coming along. Oh, that one looks pretty. So, all right. So there's DNA. Oh. I wonder if we could twist it into a helix. Oh, look. We could.
My grandma went and watched an episode of Grey's Anatomy and sat in bed and ate cereal.
There you go. 'Cause look at how long the DNA is.
Huh?
That's a long piece of DNA. I wonder what it says.
Mine's gonna get stepped on.
I wonder if we should analyze the codons and figure out what the protein looks like. Every 3 pairs, you know, from the 3' prime direction, every 3 is a codon for 1 amino acid. We could look in the book and find out what amino acid, and we could list it, and then we could build the protein. Cool. right.